So yeah, this is going to be a little different. I'm a clinical investigator, and I see my job actually, and I work in San Francisco General Hospital, and I take care of HIV-infected patients. My career and most of my group has sort of sat between the basic laboratory and the clinic. And what we've done over the years is try to take ideas from sort of basic discovery and then move them into the clinic and do first-in-human type studies, and largely to find out whether or not we can actually intervene in a way that will help patients, but also as probe studies to understand the biology of HIV. And we do that often by doing these studies and sending the blood work back to more basic scientists working on the arena. Okay, so this is not going to work, but that's fine. So our program, and what I'm going to talk about, we have a big program. It's actually growing, and it's quite virtual, involving collaborators all over the world, to be honest, that is focused on trying to understand how the virus, HIV, and the immune system interact during therapy, during antiretroviral drugs, when the virus is essentially fully suppressed, to try to take information about those virus host information and then try to come up with interventions to get rid of the virus. We want to cure people. Why do we want to cure people? Antiretroviral drugs make people live a very long time, and they restore most of patients' health. But there are limitations. The primary one is that the current antiretroviral drug regimens have to be taken on a daily basis, every day, for life. And if you're infected at the age of 20, that's 50, 60 years of exposure to potentially expensive and toxic drugs, which is not easy for many people to do. And also, as the global epidemic of HIV-infected people grows, and we have up to 70 million people with HIV, is what's the predicted numbers over the next several years, the cost of delivering treatment on a daily basis on a global level is becoming formidable and a concern. It's also quite clear that antiretroviral drugs does not fully restore health. There are persistent issues from the virus. It causes an inflammatory response, which contributes to cancer, heart disease, and so forth. And there's actually a remarkable amount of stigma that's associated with being HIV infected. It has a huge impact on patients' day-to-day quality of life. And in fact, the stigma issue, when you talk to patient groups, is the one that they're most concerned about in terms of why they want a cure. So, how are we going to cure HIV infection? The primary problem, I'm sure most of you in the room know, is that when you go on antiretroviral drugs and you suppress all virus replication, which some believe is possible, I'm in the camp that's saying it probably does not happen 100% of the time in 100% of people, but at the end of the day, the virus persists. And where does it persist? It persists in these memory CD4 positive T cells that are designed to persist for life. These cells largely reside in lymph nodes and lymphoid tissues, both in lymph nodes and in the gut. And there's a growing story that the virus actually lives within these B cell follicles within lymph nodes where antibodies are generated. And why would that be? Well, the B cell follicles within lymph nodes are actually quite protected, and the immune system can't get into these follicles. CD8 T cells in particular cannot get into these follicles, and hence the virus has found a place to live and persist indefinitely. A very central issue in my world, where I'm dealing with the consequences of treatment and the virus, chronic inflammation, is that the HIV causes irreversible damage to the immune system, chronic inflammation, tissue fibrosis, and that's where the virus lives. Really, really important point. The virus persists primarily in tissues that are not healthy and that are damaged. Now, the vast majority of people in the first generation of cure studies have gone about trying to cure HIV in a very simple manner, which is basically take a drug, force the virus out of this latent reservoir, and hope the immune system will kill it. This is so-called shock and kill. I've never been a big fan of it for one reason, and that is the following. This is a really well-known clinical study that was done in Boston, okay, in the clinical world. These are two patients, the so-called Boston patients, who were HIV infected, had leukemia, got a bone marrow transplant, had their entire immune system rebuilt with a donor immune system. That donor immune system was not infected with HIV. And these people, when they had 100% reconstruction of a normal immune system, appeared to be HIV negative. We could not find virus anywhere. They looked in lymph nodes, they looked in tissues, everything. They stopped therapy because they thought these patients were cured. Typically, when you stop therapy in the context of treated HIV infection, usually the virus rebounds within about 2 weeks. And it goes up to about 10, 100,000. T cells take a bit of a hit. You put people back on therapy, no big deal. What happened in these two guys is that one guy at month 4 and one guy at month 9, so they went a long time. No virus, no virus, no virus, healthy, 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 and then boom, the virus exploded. It went up to several million. One person got meningitis, almost died, had to get in the hospital. These people didn't know this was happening initially. So they may have been having sexual exposures with their partners, right? So this is the fundamental problem with shock and kill, is that if you don't have an immune system that will work, if you have one man standing, one virus left, it can pop up a year later, and if the immune system has not seen it for a while, it's not prepared to take care of it, it goes through the roof, causing profound clinical consequences, and that's actually the time when patients are most infectious. So it's our perspective that a cure is achievable, and it will be likely necessary to reduce the reservoir size with shock and kill, but that will not be sufficient. At the end of the day, I think the only viable pathway to a remission, to a long-term cure, is to reconstruct the immune system so that it works and can take care of the virus like it should. Now, how does that happen? We have actually in San Francisco, over the past decade or so, assembled a group of about 200 HIV-infected people in the Bay Area who acquired HIV, some in the 1970s, many in the 1980s, whose virus was then controlled naturally. These are called elite controllers. We have actually done a fair number of studies within these controllers, as have others. And when you dig down deep into trying to figure out what it is about people who control their own virus, it's CD8 T cells, CD8 T cells, CD8 T cells. Maybe some CD4 T cell help, maybe some NK cells here and there. But at the end of the day, that when you look at actually the correlates of control of the virus in natural infection, the only viable pathway to do that, and it should not be too surprising, is to enhance HIV-specific CD8 T cells in a durable manner. Now, many people have tried to do this with a therapeutic vaccine, right? Most of the vaccines, the first generation of HIV vaccines that were built to prevent HIV transmission, were built to enhance CD8 T cells. That actually has not worked out that well in the context of prevention. In fact, it seems to have done more harm than good. The prevention field has taken off on antibodies. But we, in the cure world, have fallen in love with CD8 T cells because we think it's the only viable pathway to maintain control of the virus indefinitely. And there have been dozens of attempts with therapeutic vaccines, I'm not going to go into any of these details, to control the virus with a therapeutic vaccine with CD8 T cells, and they have essentially all failed, right? Every time you give a person on therapy a vaccine, you enhance their T cells, they go up. You stop therapy, virus takes off, and things do not turn out as hoped. Now, why would that be? There are a lot of issues that exist that are preventing us from enhancing virus control with vaccines. These are some of the major factors, but our group has honed in on this one. This issue that there's chronic inflammation, chronic immune dysfunction as a consequence of the virus, particularly in the untreated state, that persists during therapy, and that if you don't fix that, the immune system won't work. And that is the central sort of conceptual model upon which we have built a rather large and growing program of interventions. Now, what are the data? Well, here's a figure from our group. There's been literally hundreds of studies that have basically shown the same story. If you take a person who is antiretroviral untreated with HIV infection and you measure an inflammatory marker, you see lots and lots of inflammation, T cell activation, CD4, CD8 T cell activation, monocyte activation, IL-6, blah, blah, blah. They're very, very consistent, okay? And it's quite high. And it's actually quite high early on. HIV does things to the immune system that no other virus does. CMV comes close, but HIV as a chronic viral infection causes a profound inflammatory response that happens early, long before immunodeficiency occurs, and is strongly predictive of cancer, heart disease, and so forth. A bad thing. You put people on therapy, antiretroviral drugs, it drops dramatically, but it rarely goes back to normal. And this delta is seen again in study after study. HIV-infected people on therapy have persistent inflammation. Not only do they have persistent inflammation, when you look at the consequence of that inflammation in terms of tissue fibrosis, collagen deposition, damage to the immune system, you can see it in basically anyone who's HIV infected. This is what, this is the blue here would be the collagen deposition in these various different, this is gut tissue, you see it's the same thing in lymphoid tissues, but you take, you acquire HIV infection, you get this profound ongoing deposition of collagen in lymphoid tissues. You put people, this is seen in untreated disease, it's seen in these elite controllers that I talked about. You put people on therapy, it doesn't get better, okay? And this is, I think, actually a very harsh environment to try to ask the immune system to work better. Now, why does this happen? Multiple mechanisms contribute to this. Ongoing HIV virus production, loss of your regulatory immune system, excess amount of CMV and other infections, treatment toxicities, and so forth. Doesn't really matter, that's not the most critical thing with this story I'm telling now, just to know there's multiple pathways that converge to result in this chronic virus-associated inflammation, all of it initiated by HIV. And the more that inflammation you have, the more immune, so if you look in tissue and in blood, in an HIV-infected person, in a cohort of HIV-infected persons on therapy, and you ask the question, okay, how much virus is there? The reservoir size. How much exists during treatment, right? It's usually about 1 replication-competent virus in blood per million CD4 T cells, probably much higher in tissues. On the order of 10 to the 9th, 10 to the 10th replication-competent virions in the body is what's predicted during therapy. You measure that. Not easy to do, but you measure that. And then you ask the question, okay, well, what is the immunologic correlates? And that's what we do in humans. It's these cross-sectional studies for the most part. And we've seen in multiple studies that the more immune activation you have, the more T-cell proliferation you have, the more T-cell dysfunction you have, the more fibrosis you have, the more virus you have, okay? Now the question is, why would that be? And here is where we have become inspired by the ongoing revolution. It truly is an amazing thing that's happening in oncology. Everything that I just said, this concept that HIV persists and resides in a very harsh, from an immune system perspective, the virus lives in these tissues, which are inflammatory, with T cells that don't work so well. That story has been played out in cancer, right? Many of the cancers are also diseased cells that exist that the immune system does not clear, in large part because they exist in this inflammatory tissues, which result in upregulation. So whenever the immune system gets turned on, 90% of what the immune system does after that is turn it off, right? Because of these issues of chronic inflammation leading to collateral damage and so forth. So the oncology model and the HIV model is remarkably similar in the modern day, right? So this is, when you look at the pathways that get turned on as a consequence of HIV-associated inflammation within tissues, we see upregulation of checkpoint inhibitors, PD-1, CTLA-4, TIGIT, a whole family of these, right, which are become quite famous because these are the pathways that are being successively intervened on in cancer. These pathways get upregulated in HIV, as does a variety of other different pathways that are ultimately quite immunosuppressive. So you have HIV lives in an inflammatory environment with this chronic immune suppression, which in theory will prevent T cells from doing what they need to do. Now in cancer, I'm not going to go into this, when you block these pathways, the inflammatory pathways and immunosuppressive pathways, you get these remarkable responses, particularly in virus-associated cancers. This crowd may be actually quite interested in this. A high number of the virus-associated cancers, which are very susceptible to immune therapy, anti-PD-1 and so forth. There's a strong overlap that I don't think anyone's quite worked out why that would be. But anyway, this is actually, and we see this in HIV infection. If you look in HIV-infected people on therapy and you look at their T-cell responses, the HIV-specific T-cell responses generally express high levels of these checkpoint receptors, all of which can be intervened. If you look in tissues, same story. If you look for various different pathways that get turned on, this is from monkeys, but we see this in humans. Some of these immunosuppressive things get turned on in tissues, which has led to the work that we do. Okay, and I'm just my goal here is just to give you a flavor of where the cure effort is going, and I think it's going very strongly toward immunotherapy, with a lot of crosstalk between oncology and HIV. Now, right now, we are very without shame stealing ideas left and right from the oncologists as well as their drugs, but a lot of these drugs were actually a lot of these concepts were pioneered here at UC Berkeley many, many years ago, and a lot of the early funding that actually led to the development of these drugs, particularly PD-1, was all actually developed for chronic infections. So there's been a fair amount of crosstalk between oncology and chronic virus infections that I think has been quite healthy. So what am I talking about? We're very interested, and we've been trying for 5 years without success to get our companies, our partners in industry to give us a checkpoint inhibitor, a checkpoint receptor blocker to test whether or not we can perhaps make a dent on the reservoir during treatment. We were all set up to do this 3 or 4 years ago with an anti-PD-1 study in the context of HIV infection. When the rug got pulled from everything we were doing, in large part because the signals that started to emerge in oncology were so dramatic that the companies, MedImmune, BMS, Merck, all the companies that were interested in chronic infections dropped chronic infections and put all of their resources into oncology, which makes sense, right? I mean, there's a much greater need there. So we have this entire program of HIV cure has really been on hold. What we're doing now is actually taking people with HIV who get cancer, many virus-associated cancers, and are now being treated with these various different modalities to try to ask the question whether or not we can intervene with these drugs to have an impact on the reservoir size. And those studies are ongoing, both in cancer and in the context of treated HIV infection. And what they've shown, and here's a study that was done in the AIDS clinical trial group, what they have largely shown that if you take people on long-term therapy, and this is monkey work as well as human work, and their virus is shut down, and you block PD-1, which is upregulated on CD8 T cells, you get an enhanced number of CD8 T cells in the blood. You also get evidence that the virus is actually becomes more expressed, perhaps as a consequence of the immune activation associated with blocking PD-1. And there's some evidence in monkeys that when you stop therapy in that setting, the virus is controlled. Those studies are ongoing. We're doing a bunch of studies in the context of HIV and cancer. We've also, and again, I tend to get a bit grandiose here. I'm about to get far more grandiose. There's not only this work going on in oncology with all these anti-inflammatory drugs, there's a massive amount of interest in blocking chronic inflammation, some of it related to chronic infections, particularly CMV, in the HIV negative population who are getting older. Right? There's a big story that aging, the aging process, getting old, is driven in part by chronic inflammation, and that this is driven in part by chronic virus infections. And the National Institutes of Aging have a huge program trying to come up with drugs that prevent aging, the most powerful and interesting of which is rapamycin, sirolimus, which is an MTOR inhibitor. Okay, now why am I saying this now? So that whole line of investigation has also inspired us, because by blocking mTOR in people, you reduce inflammation, which makes it easier to generate memory T cells. We are doing those studies as we speak in these early experimental type medications. And when we've actually have some cross-sectional studies from work that we've done in the past suggesting that by blocking mTOR inhibitors, by blocking mTOR, you enhance T cell function, you reduce T cell activation, you generate better memory, and you reduce the size of the reservoir. We're doing that. We're doing this with JAK-STAT inhibitors. We're doing this with inhibitors of IL-1. And again, I'm just here to give you a flavor of the kind of work that we're doing. And so there's just a growing number of studies that are aimed at reducing these inflammatory pathways, with a lot of our ideas coming from oncology as well as from aging. And that, we think, will result in an environment that will allow T cells to work better. So that's one big pathway. The second big arena is, okay, so you make the immune system more healthier, less harsh. Now you got to actually vaccinate people. You got to enhance the immune system. And there, actually, we and others have these big programs looking again at vaccines, which have not worked that well in the past, as I said earlier, in large part because I don't think that they were given in a proper way. And another big exciting area that's being driven by Gilead, the platinum sponsor of this meeting to a great degree, is to enhance the capacity of the immune system to work better by just making the T cells work better directly with T cell adjuvants. Gilead gave a bunch of monkeys a TLR7 adjuvant, which they had developed for the treatment of hepatitis B. Didn't work, but they gave it to these monkeys and showed that by blocking by stimulating TLR7, you get this adjuvant effect on T cells, and T cells work better. You stop treatment in the monkeys, and the virus is controlled. So there's this whole other pathway that we're working on. And in fact, over at UCSF, we just got a $20 million grant from MTOR, a foundation, largely to develop from the laboratory into people, manipulating TLR pathways to make the immune system work better so that we can achieve this remission. Finally, vaccines. Okay, I just want to throw out there the hint. So the other thing that our group is most excited in, so we have this three-pronged approach to curing HIV infection. We're going to enhance the immune system's capacity to work, make T cells better with vaccines, and then of course we have to vaccinate people. And I'll just say that there's this fascinating story that's come out of Louis Picker's group that we're working very closely with him on, where he's taken CMV, he's reconstructed CMV, he's rebuilt CMV, very inflammatory virus, so that delivers SIV and presumably HIV proteins to the immune system. He has shown in that setting that if you vaccinate monkeys with a CMV vector, you generate these whopping CD8 T cell responses that does not prevent SIV from getting into the monkeys, but results in resolution and clearance and cure of those monkeys subsequently. So the UCSF, MTOR, NIH, DARE, Gilead, combination of various different collaborators. We have a very big program right now, 4 or 5 different distinct pathways going from the laboratory into the clinic. And I'm here in part to just inspire people that this is the kind of work that we're doing, but also in these first-in-human studies, we're collecting a massive amount of biologic specimens that we often like to share with people who are interested in helping us figure out these riddles in a relevant setting, which is typically what's happening in the context of these clinical interventions within people. So with that, I'll stop and acknowledge some of my colleagues, Mike McCune, Peter Hunt, Sharon Lewin, Danny Dewick, Rafiq Sekli, Nicholas Tremont, Louis Picker, and a bunch of other people who are showing up here have been central to the work that we've been doing, and happy to take any questions. Thank you.
Hello. Looking at the elite controllers, have you found any evidence of altered homing of CD8 T cells?
Altered homing?
Yeah.
Altered homing.
I mean, can they penetrate the follicle at all?
No. So actually, it's interesting. In the elite controllers, in monkeys, and there's a paper coming out in humans as well, that you can't find the virus really in any tissue, except in the B cell follicle. And the CD8, these very powerful HIV-specific CD8 T cells, they come up to the follicle and they just sit. They provide this like barrier. So the virus replicates at will within the follicle, which is not good for the immune system. That's probably why there's all this chronic inflammation. But as the virus leaves the follicle, it gets killed by the CD8 T cells, so you can't see it systemically. So the idea there, in order to try to reverse that, is that we're trying to come up with ways to break down the follicle, which you can do with, Louis Picker and Jeff Lifson have been doing this by breaking down the follicle with anti-CD20 with rituximab, which basically results in a follicular breakdown, which allows the T cells to get in and presumably clean up the virus.
So thank you for the very insightful presentation, and it's a very interesting concept. And you said you are not a big fan of the shock and kill, and indeed, a major problem of shock and kill is that it's very inefficient, and several people reported that a single dose of the shock and kill drug, a histone deacetylase inhibitor, can only reverse latency in 0.47% of the cells, of all the cells latently infected. So compared with shock and kill, how efficient is your immunotherapy strategy? And if it's making into the clinical trial, how long do you expect the therapy should persist in order to clear all of the latent reservoirs?
Right. No. You great point. So the current collection of shock drugs, HDAC inhibitors, barely just tip of the iceberg. They're not really doing much to the reservoir size, in my opinion. Others may disagree. But that's the magic of the approach that I just outlined. You don't necessarily need to reduce the reservoir. What we're looking for is control, not reduction. So we have assumed that the virus is not going away. It's going to be there for life, and you just need to control it. Now, the work in elite controllers and in other settings suggests that the amount of virus that you actually can control with the immune system is about 2 logs lower than currently happens in people. So I would actually think that we are going to need to reduce the reservoir, probably by about a couple logs, about 10 to 100-fold, with presumably shock and kill. And that once you get there, then just like with cancer, if you can reduce the overall disease burden, the immune system works better. That's the idea.
Hi, Steve. I was just wondering, after radiation therapy, for example, what was done in the Boston patients, what happened to the T follicular helper cells in the lymph nodes? Are they destroyed, or do they remain? After
radiation therapy for cancer and so forth?
Yeah.
It's interesting. I found from a cultural perspective that you're asking a very fundamental question that could be asked in people with cancer, but no one's ever really asked that question as far as we looked. I don't think oncology in general asks those types of questions, or just the pathogenesis is not something that's often asked of people. The people are sick. So I don't really, we've looked for those kind of data from the oncology world. It probably exists, but I don't know the answer to that.
Have you done any work with ex vivo vaccination in order to enhance the cells that are most directed before the patient sees the immunogen?
Have I? No, I'm a clinical investigator, and so, but we've done when you say there's a lot of work that's the SIV model in monkeys is extremely well validated. So most stuff goes through the humanized mice and into the SIV model. Looking at the capacity of immunogens to enhance T cell responses in vitro, I think is clearly people do that, and that's where you get your hints from, but our group doesn't do that.
The approach that you've described requires addressing simultaneously multiple approaches, addressing multiple mechanisms. carrying out a clinical trial that addresses multiple aspects is logistically and from a regulatory standpoint complex. Are your trials designed in a way to do that? And if so, how?
Well, the regulatory aspects of clinical trials is out of control. It is a big issue. There's multiple people who are involved in this, and it can suck the soul out of a clinical researcher, but you just have to sort of bear with it. It is doable. It's feasible. You have to have a fair number of resources for all the regulatory stuff. And so we've been able to do this. We have found that the FDA in particular has been very friendly toward this type of work in HIV cure. They see it as a the main reason why I think that we've been able to do these studies is that because they're very intensive, there's a fair amount of risk, there's a fair amount of unknowns, and the immune system is a big black box, and you're really not sure what's going to happen. Is that we are blessed in the HIV world to have a group of people, HIV-infected adults, who are just generally very educated, very motivated, very community-oriented, and often will do things that are out for altruistic reasons. So these studies are a huge burden on the patient with a fair amount of risk. And we've never had a trouble in terms of getting community support, patient volunteers, or regulatory support. And I give a lot of credit to the community that work, and it's quite distinct and quite unique in HIV. So and each of these studies are actually very pilot exploratory. And so what we're trying to get from each of these different studies is a signal, and then we figure out how best to combine them in the future. Most of these combinations are being studied first in monkeys, and that's how we do it.
I think I understand the logic that you had with the humanized monoclonal against PD-1 or CTLA-4 is that if you have latent infection and you get rid of the brake, you now attack the cells with more robust energy.
Right.
But the data that you showed, I was confused. That what seemed to be happening was that you're actually getting more viremia.
Yes.
So can you explain that?
Oh, yeah.
I mean, I love the logic. I just don't understand the data.
No, I chose to stay big picture here. But you asked a great question. So HIV is enriched in, so HIV enters a memory CD4 T cell. That memory CD4 T cell becomes activated. It dies. Some don't. The ones that don't die often express PD-1. So PD-1 is now a marker of an HIV-infected cell. You block PD-1, you activate that cell. It becomes shock. So you would expect, by blocking PD-1 in particular, that the CD4, that you would actually be turning on CD4 T cells enriched with HIV. Those CD4s become activated, you make virus. And in fact, that's what we're seeing. And that's a good thing, because that's shock. So it's a beautiful approach, right? You shock the virus, and Nicholas Chamont has a lot of data that's being put together on this particular very important study. But you block PD-1, your CD4s actually get activated, virus comes out, at the same time you're enhancing the kill. Shock and kill. And that's what we're seeing. If you get in the cancer, you give a PD-1 blocker, you get an increase in cell-associated RNA. You're making more RNA in cells, but the amount of viremia over time drops. So the amount of virus that actually gets released drops over time because you're killing the reservoir. That's the theory. Yeah. Thank you.